Hey everybody, let's talk a bit about stem cells and a few other things. In this next set of lectures, we're going to look at stem cells, talk a little bit about cancer, a little bit about viruses, just a few kind of not random, but just topics that don't fit into the rest of the course very well that I would like to cover so you have a good overview of biology before you go. Stem cells. I'm sure you've heard of them. Stem cells are unspecialized cells, meaning they aren't liver cells, they are not brain cells, they're not bone cells, but they can divide indefinitely. That's one of their traits, one you should know. They can divide indefinitely. What does that mean? That means that their telomeres don't get shorter as they divide. That means they add more telomeres. They have telomerase in their cells, so their telomeres don't get short. They can keep dividing, and they can differentiate into more than one type of cell. And so here's an example of that. Here's a stem cell, and it can divide to make more stem cells. It can also divide to make a progenitor cell or several kinds of progenitor cells. That progenitor cell will receive various chemical signals to cause certain genes to turn on or off, which will then lead it to divide into one or more kinds of cells. So a progenitor cell might be a little less free to divide into lots of different cells than a stem cell will be. It's a little more specialized. It can, in this case, this kind of progenitor cell can become a fat cell, a bone cell, or a white blood cell, depending on what chemical signals it receives, what it's told to do. Okay, so in this example, this stem cell is a stem cell derived from bone marrow, and it can divide into any one of these things. So the progenitor cell is still a kind of stem cell. It's more like an adult stem cell. It can divide into several kinds of cells. It's just not as free to divide as like an embryonic stem cell would be, a stem cell that could become any kind of cell in the body. So early animal embryos have stem cells that are pluripotent, meaning they can become any kind of cell, kind of like that hypothetical stem cell in the last diagram. And they can reproduce indefinitely. The adult body also has stem cells, which are more like this progenitor cell here. They can divide, and they don't just divide wherever. They replace cells that don't divide on their own, like making new red blood cells. Red blood cells don't divide by mitosis. We make new red blood cells using our stem cells in our bone marrow. And because working on embryonic stem cells has some ethical implications and it's hard to get a hold of, a lot of research has focused on these adult stem cells. And they've been trying to find ways to make adult stem cells more pluripotent, which means you would take an adult stem cell and modify it to make it more like an embryonic stem cell that can become any kind of stem cell. It may become more clear in this diagram. So on the left, we have an example of a human embryo. So remember, an embryo forms when a sperm meets an egg and we have fertilization. We make a zygote. It's a diploid cell. That divides by mitosis and to make an embryo. So once that zygote has divided, it's an embryo. If you harvest cells from an embryo and put them in a petri dish and grow them and put them in different kinds of conditions with different chemicals and things, different chemical signals, ligands, if you will, they will develop into any one of these kinds of cell types or more. So liver cells, nerve cells, blood cells, they're pluripotent. They can develop into any kind of cell. In an adult, we find stem cells, but they're a little more specialized and they are limited. So if you harvest cells from bone marrow cells can only develop into different kinds of blood cells, for example. They can develop into more than one kind of cell, but it's within a limited range. And so one of the goals of research is to try to make these cells behave more like embryonic cells. And there has been work done on that, trying to deprogram differentiated cells and make them act like embryonic stem cells, which is what ES stands for. And they've been working on this. In fact, a Nobel Prize was given for this work in 2012. Now, another thing I want to mention then is cells develop, they divide, they follow chemical signals. Cells even have a way to self-destruct if they misbehave. It's called apoptosis. You can read about it if you wish. It's really interesting. Apoptosis is why we have fingers that are not webbed. When we were embryos, we had tissues between all of our fingers, but that tissue self-destructed before we were born, typically anyway. Now, my dog is a black lab. He still has webbed fingers. He can swim really well, right? His toes are webbed. That's when those cells did not self-destruct. But there's all kinds of the soup of chemical signals, ligands that dock to receptors that cause transduction pathways to kick on, and different transduction pathways to kick on, and different cells leading to different things turning on and off, and development to take place. And the same systems that control development lead to cancer when they go wrong. All of these chemical signals lead to bad behavior when something is off. And research in cancer-causing genes have led to the discovery of these oncogenes, which are genes that can lead to cancer. There's very specific genes that we know when something goes wrong, it often leads to cancer. And what often happens is when you mess with one of these genes, it leads to extra proteins being made or incorrect proteins being made, something happening that releases a bunch of proteins that shouldn't be there or that cause some kind of problem in cells to behave incorrectly. So here's an example of one of these genes. This is one that's a proto-oncogene is a normal gene, right? And here it is doing its job. This is the gene. There's the promoter. This is where RNA transcriptase would dock, and it would copy the gene, and it makes these proteins down here. These are growth-stimulating proteins, and that's what it's supposed to do. But one problem that might happen is we might copy that gene too many times, and then we get extra growth-stimulating proteins. Growth-stimulating proteins might cause a cell to divide by mitosis over and over and over and over and over and over again, right? That's a problem. Another example of a problem might be some issue with the gene, maybe a mutation in the gene leading to extra proteins being produced, or you might have a mutation in the gene that leads to a protein with the slightly wrong shape. Actual growth proteins are usually made, they do their job, and then they're degraded, they're broken down. But maybe this protein is hyperactive, meaning it really works too well, or it can't be broken down, so it causes mitosis to happen over and over and over again, even when it shouldn't. So these are issues that can arise that make us lose control. Cancer typically arises when we lose control of the cell cycle. Here's another example of how cancer might arise. So here's a receptor, a growth factor docks, like we talked about. It causes a transduction pathway to kick on, starting with this molecule here that is activated by a G protein, right? And then it activates a protein kinase chain, so lots of phosphorylation, that turns on transcription and leads to the production of a protein that turns on the cell cycle, right? That's normal. But sometimes mutations happen. This protein here suddenly doesn't need, look, there's no signal here. Sometimes mutations cause this whole thing just to stay on. The RAS protein here has mutated, and it doesn't have an off switch anymore. It can't be shut off. It's always on, and it just constantly produces growth stimulating hormones essentially all the time, and that's how a tumor might form. It just grows and grows and grows and grows and grows. Mitosis doesn't stop. We're constantly growing and dividing, and that's one of the ways we might end up with a tumor forming. It's an interesting chapter here, chapter 16. You might want to check it out for further information. I just wanted to touch on that to tie it back to the cell communication stuff and mitosis that we talked about earlier in the semester. The last thing I'm going to lecture on in the next video will be viruses.